Hello dear students, welcome to the session. We are going to discuss 10 questions on molecular basis of inheritance. So let's begin. The first question you can see, DNA fragments are negatively charged, they are neutral, they are positively charged as well as negatively charged, depend on the size, or they are positively charged. What is the answer here? Okay, you have learned this concept that if you talk about DNA is made up of nucleotide, right? It's a polynucleotide, right? And in nucleotide, what you have three things, right? You have first thing, that is sugar, right? Second thing, you have a nitrogen base, okay? And third thing, which is responsible for imparting negative charge to the DNA, is phosphate, right? This gives negative charge to DNA, right? So DNA fragments will be having which kind of charge here? Negative charge. Option number A will be the answer, right? Second question. Which of the following RNA should be most abundant in animal cells, right? So for solving this question, you require one particular concept which is called polysome or polyribosome, which you have learned in prokaryotic cell. Okay. What was this concept? This concept says that if you have one messenger RNA, right? If you have this one single messenger RNA, right? Definitely your messenger RNA will be having start codon, right? It will be having next to three codons, next to three bases means one codon, again next three, again next three, and at last you'll be having UAA or any three of the stop codon can be there, right? Up the thing is that first ribosome will come and bind on this start codon, right? And it will begin the process of translation, okay? And you know that the ribosome moves three steps at a time, right? So here by this time you will see 1, 2, 3, right? 3 amino acids are already added in this growing polypeptide chain, right? Again, this is free or not? Again, one more ribosome can come and bind here and start doing, you can say, protein synthesis, right? Again, this will move further, okay? This one will move further, okay? So like this, if you have one single messenger RNA, right? Let's suppose here also, or let's suppose one first ribosome which binded, I'm making drawing here. So this first ribosome, this is the second ribosome, this is the third ribosome, but you have only one single messenger RNA, right? And here till here, it will be having 1, 2, 3, 4, 5, right? 1, 2, 3, 4, 5 amino acids. Here 3 amino acid, here only 1. Okay. What you understood from this? For this, again and again, you will be requiring transfer RNA. Transfer RNA will come and bind here and will attach the particular amino acid, will bring the particular amino acid. But you remember, transfer RNA can leave this and again they can get amino acylated. So transfer RNA is not needed that much, right? Ribosomal RNA is needed in abundant of the amount. Why? Because you can see from one single messenger RNA, you are able to make so many proteins, so much protein, right? From one single messenger RNA, you are able to synthesize lot of protein, right? And tRNA can be again amino acylated, so you don't want them also, right? But you require lot of ribosomes, okay? And if your ribosome requirement is more, okay, ribosome requirement is more, right? So you require the structural RNA for making ribosome or not. What is the name of that structural RNA which makes ribosome? It is ribosomal RNA, right? So what is the answer? rRNA. miRNA is not in our syllabus, right? It is also used in some regulation processes, gene regulatory processes, but they are not in our syllabus, so we don't have to discuss about that. Okay, tRNA, mRNA and ribosomal RNA mein, what is the condition? The you can say abundant may what you require in any kind of cell, not even animal cell, in any cell you can consider. First, you require a lot of ribosomal RNA. After that, you require like close to 15% you require transfer RNA. And after that, very less you require messenger RNA, right? This will be the sequence of how much abundance type of RNAs are required in animal cell during protein synthesis. Okay. Then uridine present only in RNA is what? Is it a pyrimidine? Is it a nucleoside? Is it a nucleotide, or is it a purine? So you have learned some names like uridine, cytidine, right? Uh, Guanosine, okay? Adenosine. What are these names? Okay, so you know there are terms like nucleoside and nucleotide. Okay, if I'm writing here adenosine, okay, if I'm writing here guanosine, if I'm writing here uridine and cytidine, okay, that means I'm talking about what? I'm talking about nucleoside. What is the meaning of nucleoside? Who will tell me? Nitrogenous base plus sugar. If I attach a phosphate also here, I'll be able to make a nucleotide, right? So what I will write? I will write adenosine monophosphate. Okay, this will be the name of nucleotide. Guanosine monophosphate, uridine monophosphate. This will be the name of nucleotide. And one more thing I would like to tell you. If you just want to see what is the name of nitrogen base, right? So if I consider only base, you will call it as adenine. Here, guanine. Here, uracil. Here, cytosine. If you attach a sugar also, the name will change. Okay, if you will attach a phosphate also, the name will change. And these are the names for what? These are the name for RNA, right? I have just written everything which is there in RNA. If I want to write for DNA, I will be writing deoxyadenosine, deoxyguanosine, right? Because the sugar is different there. So now you understood what is the meaning of uridine here. It is a type of nucleoside, right? It's a type of nucleoside. Okay, it's not pyrimidine. Okay, it is a purine, right? But it is made up of purine. Uracil is a purine, but if you attach sugar, it will become uridine and you will call it as nucleoside. Understood? Okay. Now, if one strand of DNA has a nitrogenous base sequence of A, T, C, T, G, what would be the complementary RNA strand sequence? Very easy question. They just have given you one sequence, right? And they are asking complementary, right? They are asking complementary RNA sequence, right? And they have not defined that whether this particular DNA strand which they are writing A, T, C, T, G, this is 5 prime to 3 prime or 3 prime to 5 prime. They just mentioned that this is one of the DNA strand. Okay, this is one of the DNA. You have to tell the complementary to make RNA. Okay, it means that this is template strand. Right, this is template strand. If you are considering this as template strand, what is the complementary of this? You can write 3 prime to 5 prime. That is no problem, right? Then if you want to make RNA from this, instead of A, what you will write here? U, right? Because thymine is absent in RNA. Here you will write A. Here G. Here what you will write? Here you will write A. Here C, right? So this will be the sequence of your RNA. What is the answer? U, A, G, A, C. Understood? Okay, this question. The unequivocal proof of DNA as a genetic material came from the studies of, remember which were the scientists who gave this unequivocal proof? This is a PYQ, very favorite PYQ. I think 2 times they have asked this question, right? After 2021, you just check, they have given this question 2 times at least. So what is this? This is the experiment which was done by Hershey and Chase, right? Alfred Hershey and Martha Chase, they have done one experiment, okay, and they have understood that DNA is acting like a genetic material in which organism? Was it a bacteria or it was a virus? So it was a virus, right? They have taken host cell as E. coli, which was a bacteria, right? But they have taken what? They have taken, you can say, bacteriophage. What's the meaning of bacteriophage? It is a virus infecting bacteria. Okay. And they have taken which type of bacteriophage? I will write the name. It is not given in NCERT. T2 phage. Right. So actually they wanted to know what is the genetic material of this virus. Okay. which is infecting a bacteria. So can I call this virus as a bacterial virus? Yes, right? They have done an experiment in which they have understood that DNA is acting like a genetic material of this virus, because this T2 bacteriophage has genetic material which is double-stranded DNA. And to make more viral particle, it was injecting its DNA inside the bacterial cell, and as a result, more and more viral particles were getting formed. So it means that what is acting like the genetic material here? Okay, DNA. And they are checking that DNA is acting like a genetic material in which organism? Bacteria or virus? A virus that infects a bacteria, right? That is bacteriophage. Which is bacteriophage is used? T2 bacteriophage. What is another name of this group of bacteria which infects this group of viruses which infect bacteria? It is bacterial virus. Okay, understood. Now, which one of the following does not follow the central dogma of molecular biology? That day I discussed something about this, right? If you remember, I have discussed with you that there is one concept which is called reverse central dogma, which is just you can say genetic information flows in a reverse order, right? So that happens in some of the viruses. So I will mark here HIV. You just see that these all, if you consider, they are at least they are organisms, right? They are at least cellular. This is a common character. And HIV is what? This is non-living. This is acellular, no cell-like structure, right? And I told you all the organisms, they follow central dogma of molecular biology, except this. What they follow? They follow reverse central dogma, right? They follow reverse central dogma. Okay, so viruses, they carry their own enzyme which is called reverse transcriptase, by which they will allow the flow of genetic information in a reverse order. From RNA, they will be making DNA, right? Whereas mucor, what is mucor? Mucor is fungus, right? It is belonging to which group? Please write in the comments. Mucor belongs to which group of fungi, right? Okay, it belonging to Phycomycetes or Deuteromycetes or Basidiomycetes or Ascomycetes? Please write the answer. Mucor belongs to which group of fungus, right? And Chlamydomonas, according to plant biologist, what it is? It is green algae. According to R.H. Whittaker, what it is? It is Protista, right? And P belongs to which family? P family. Please write this also in the comments. What is the family of P? Right? Very easy. Please write. The two polynucleotide chains in DNA are. Okay, this question just see. If you consider the two polynucleotide chains of your DNA, right? This is one chain, this is another chain, right? What are the characters of this chain which you can consider here as per given information? Okay, they are, both are discontinuous. No, only one of the strand is formed in a discontinuous manner, later it is joined, right? So we cannot choose this. Okay, both are semi-conservative. No, entire DNA is semi-conservative. Both the polynucleotides are not semi-conservative. One is parental, one is new, right? So we cannot choose. Parallel, no. Parallel means same polarity. 5 prime to 5 prime. If this strand is, another strand should also be 5 prime to 5 prime. No, this is also wrong. What is the answer? Anti-parallel. Sorry, anti-parallel, right? You can see this is 5 prime end, and opposite to this, what is there? 3 prime end. Okay, same condition there also, right? You don't have 5 prime to 5 prime. This condition never exists. Always remember, you can never make like this 5 prime, 5 prime, 3 prime, 3 prime like this. This is wrong, right? It is not possible. Okay, and I have told you reason also, right? The direction of synthesis is different. This strand is formed in this direction, right? Arrow you can see, and this strand is formed in this direction, right? That is why they are 5 prime to 3 prime anti-parallel to each other. Fine. Okay, which form of RNA has structure resembling to clover leaf? Okay, clover leaf is like this. When you will see a clover leaf, you will see it is having 4 ends, right? So this kind of 2D structure is there in transfer RNA. The 2D structure of transfer RNA is clover leaf, and the 3D structure is inverted L shaped, right? Clover leaf structure, actually it looks like this. If you will check the 2D structure, it is also having 4 arms like this, right? Like this, 4 you can say, you can see the leaves, right? So like that it is giving this name, and here to this end amino acid is also attached. Okay, this is 5 prime, this is 3 prime end. So it is like clover leaf shape in 2D. This is 2D structure. Okay, this one. Fine. Now, reverse transcriptase is RNA dependent RNA polymerase. Okay. So the thing is that reverse central dogma of life, if I discuss, okay, if I discuss about reverse central dogma of life, you will study there. They will be having this information like this: DNA can make DNA by replication. DNA will be able DNA will be able to make RNA. RNA can make protein, right? But these are the viruses where the main genetic material, okay, these are the viruses where the main genetic material is RNA. Okay, like tobacco mosaic virus, like HIV virus, right? Retroviruses, they all have RNA as their main genetic material. So when RNA is the main genetic material, then how they are going to replicate when they will enter in the host cell, right? From RNA we can never make again RNA. So what to do? So the information which they have, it flows in reverse order. They are having like they have proteins, right? Definitely these viruses are made up of genetic material plus protein. Two things are there, right? One of that protein, okay, RNA, when it will enter inside the host cell, it will start making protein. One of that protein will be reverse transcriptase. Reverse transcriptase, this is the name of that protein. Okay, this reverse transcriptase will allow this reaction where you will be making DNA from RNA. So who will act like a template? RNA will act like a template, right? And what you are polymerizing here? DNA. So can I say that reverse transcriptase is a RNA dependent DNA polymerase? Yes. Okay, it requires RNA as a template and it will be making the DNA on that, right? So it is polymerizing deoxyribonucleotide. It is a DNA polymerase and it is using template as RNA. So it is RNA dependent DNA polymerase. Understood? Okay. Now this one. What is not true for genetic code? This is very easy question and you can solve it very easily. Just see that last statement. Codon in mRNA is read in non-continuous fashion. Is this correct? This is absolutely wrong and you have to find what is not true. Okay. When you will check NCERT, they have mentioned code, genetic code is universally nearly similar except for some codons in some protozoans and in mitochondria some codons vary, right? Rest all, every triplet will code for same amino acid in everyone, right? It is degenerate, it is unambiguous, right? One codon will specify only one amino acid and one amino acid can be made by multiple codon, right? So these all things you have learned in genetic code. Okay, this was the last question. So these were the 10 questions for today. I hope you have understood clearly. If any questions or any doubts are there, you can write in comments. Thank you very much. Bye.